Our next speaker is Dr. Federica Brandizzi, who is a plant, cell, and molecular biologist. She joined Michigan State in 2006 as an associate professor. She's currently an MSU Research Foundation professor and MSU Distinguished Professor. She's also, as Bruno mentioned, the chief scientist of the Great Lakes Bioenergy Research Center, and she has a very large research program. The long-term goal of Dr. Brandizzi's research is to understand how plants grow and interact with the environment from a subcellular perspective. Dr. Brandizzi's work has contributed to the understanding of how organelles of the plant secretory pathway establish their morphologies and functional identity, and how this knowledge can be applied to improve crop productivity. Her work has been published in more than 180 publications. Please join me in welcoming Dr. Federica Brandizzi to the stage.
Good morning, everyone. How are you doing? Are you excited? Yeah, I'm so excited and so grateful and to be here today to spend the morning with you. And I feel this is a privilege to be able to show research and what we do at Michigan State. And Michigan State is a fantastic place for a person like me that loves plants, that wants to work with plants and wants to understand them from the subcellular level, like Phil was saying, and use that knowledge to improve plants and allow better lives for everybody on this planet. So it's a big goal. Let's see how it goes. Yeah, we want to improve plants for a better future. I have 3 children. They changed my life in so many ways, and I really want to leave a better world for them and for their own children. So let's see, where do we start? Because when I look at predictions, the future is not bright, okay? When we come to prediction, are we going to have enough food to serve the world population that is growing at an incredible pace? So let me see if I get this right here. I usually mess up with these things. No, I got it. So this is where we're gonna go by, almost 10 billion people by 2050. 2050 is around the corner, right? So do we have enough food for them? No, unfortunately not. So at the growth rate of the world population and the food that we can put forward, we're not gonna match what people are gonna need. So we need to improve plants. And why? Why is this important? Well, plants, of course, provide food, but they also provide material for construction. They also provide platforms to produce medicines. Did you know that a COVID vaccine was produced in plants? I didn't know until I had to prepare this presentation, actually, but it's true. And also, plants, because of their biomass, can be converted to biofuels and bioproducts. For example, did you know that the plastic bottles that you can use for drinking water come from plants? The plastic is produced from plants? Well, I didn't know. And now I didn't know until I had to study. So, but let's see. But who's the mastermind behind all these things that plants can produce? It's actually a set of organelles that is called secretory pathway. Okay? The secretory pathway is made of different organelles, so tiny compartments that live inside of a cell. So let's imagine that our body is a cell, our organs are the organelles. So subcompartments, they are doing different things. In the secretory pathway, the mastermind is an organ that is called the endoplasmic reticulum. And this one produces lipids, so fats, proteins, but also some forms of sugars that then are transported to a Golgi apparatus. Golgi, you may recognize as an Italian name, the guy that discovered, just putting it there. And then endosomes and then a large vacuole, and then there is the plasma membrane, which basically envelopes the entire cell. Now, the secretory pathway is essential for plants and humans and anything that feeds on plants and breathes air, because basically by functioning, the secretory pathway allows life on this planet. So produces the food. The food is usually stored in the storage vacuole. Then the secretory pathway produces enzymes and compounds that are put into the cell wall. So every plant cell is surrounded by a cell wall, and the cell wall essentially makes the material for construction that we use. But the secretory pathway is also important for protecting plants from attack from pathogens, because it produces antimicrobial peptides or proteins that are secreted outside the cell, and microbes don't like those. So it's a really an important set of organelles. As I was mentioning earlier, essentially we can live because of the activities of the secretory pathway. The plant endoplasmic reticulum feeds the world with the seed storage proteins that it synthesizes. This is a famous statement from two pioneers in the field. The secretory pathway can produce vaccines and very important molecules that we can force it through molecular biology, we can force it to produce and produce precursors for biofuels and bioproducts. But just to make an example of, that can be core at our heart. So we all familiar with proteins. They're important to build up the cell and the entire organism. And there are different kind of proteins. And I want to talk to you about one that, of course, we find in pasta, not because I'm Italian, right? But it's gluten. And some people are allergic to gluten, but for the rest of, of humankind, gluten is very important. It is basically produced and stored in seeds, like wheat seeds, for example, but is produced from the endoplasmic reticulum, and is very important, of course, to make food for us. Now, in my lab, we're trying to understand how the endoplasmic reticulum can be improved to produce proteins like gluten and other very important food precursors. But also in my lab, we're trying to understand how the secretory pathway can produce antimicrobial proteins. And I'm very proud to say that for decades, the field thought that there was only one biosynthetic route to produce antimicrobial peptides, but thanks to the resources that Michigan State allowed us to use, we were able to identify a new biosynthetic pathway, and this has been published in a prestigious journal. I'm very proud to say that because it was a major effort in our research. But now I want to give you a deep dive into a plant cell. So far, I showed you diagrams that are more or less beautiful to look at. So I want to give you the reality of how the secretory pathway looks like in plant cells. So let's deep down. This is a plant. This is our model plant. It's called Arabidopsis. You can find it in your backyard. It's a weed, so it's not really nice to see. But anyway, let's try to look now with a microscope on the surface of the plant. On the surface of the plant, like on our skin, we have the epidermis and the epidermal cells. So this is how an epidermal cell of a plant cell looks like. It's very similar to human cells. Now, if we look inside the cell, we now look at the endoplasmic reticulum, the Golgi apparatus, and the vacuole. I would like to ask whether we can dim down the lights, if possible, at the front. If not, not to worry, but I think the contrast would be nicer. Yeah, can you see it better? But they just don't look like that. They actually move. So let me show you. Look how the endoplasmic reticulum is moving. Now, the Golgi apparatus is moving too. You bet the vacuole is going to move? Yeah, the vacuole is moving too. So there is so much life inside plant cells. Actually, this is real time. So when you eat the salad, that's what you're eating. This is the life you're eating. And it's beautiful. It's absolutely beautiful to look at. Because thanks to, again, I mean, this sounds almost an advertisement to Michigan State, but I had to say this. Thanks to the microscopes that we have here, we can look at organelles in real life, but also look at how these organelles interact with each other. So let me show you here. Ooh, can you dim a bit more, please, the light, if possible? So that those that want to go to sleep, I will not see you, but thank you. I'm so grateful. I really appreciate that. So look at the Golgi apparatus, and now look at the endoplasmic reticulum. Underneath, the Golgi is moving on the endoplasmic reticulum. And I want to give you an idea of the size of what we're talking about. A Golgi stack is half a micron in diameter, and a micron is 1/millionth of a meter, or 1/25,000th of an inch. Sorry, my accent is terrible. Anyway, now with microscopes, what we can also do is to check, and this was a major breakthrough that my former postdoctoral advisor did, that these two organelles interact with each other. So what Chris Hawes, late Chris Hawes, did, he blocked the movement of these organelles using a drug that kills the cytoskeleton, so the skeleton that allows organelles to move. And what he did, using optical laser tweezers, he grabbed a single Golgi and he moved it around the cell. I want to show you this video again because it's just mind-blowing. So you can take this little organelle and move it around the cell. That's fascinating. I love it. So now, there is so much that we can learn from other fields. The mammalian field is more advanced than the plant field, blah, blah, blah. But we cannot usually use the information that is available from mammalian cells or even yeast cells and translate them to plants. So this is a plant cell. This is the endoplasmic reticulum, the Golgi apparatus on top. This is a mammalian cell. Can you tell the difference? This is our cells. The Golgi apparatus is here next to a big nucleus sitting on the endoplasmic reticulum. And the mammalian Golgi is lazy. It doesn't move, okay? So there is so much that we can learn from plants. And for example, my lab helped understanding how these organelles move. So in every cell, there is a cytoskeleton. Like in our body, there is a skeleton. And in plant cells, the skeleton is mainly actin-based. Also, our cells, mammalian cells, have actin. And plant cells use this formidable cytoskeleton to move organelles around. You can see that, for example, the Golgi apparatus is sitting on the actin cytoskeleton and moving around. So my lab has also made an important discovery as time progressed, that there are proteins that allow tubules of the endoplasmic reticulum to fuse with each other. So you can tell here there is so much life. You can see the tubules forming and then fusing. So I want to show you, as the video restarts, I want you to put attention on this tubule that is coming down. I don't like it. And then he tries again. I like it here. Okay? So my lab identified, thanks to an extensive genetic screen that we did, a protein that is called RHD3 that is important for the controlling and enabling of this fusion. So you may ask, okay, we're talking about sustainability. How that, studying these details, apparent details, can impact the bigger science? And I want to show you just an example. So we learned that throughout cell development, so as cells expand, so we're looking at now leaves that become older and older, so from 3 days after germination of little seedlings to 12 days, that the endoplasmic reticulum in a wild-type plant changes its shape from very sheety, not how I said it. Okay, I was, I got caught once in a big meeting for saying, okay, never mind. So, so from here, going through this very nice reticular structure, now you can see that in a mutant that lacks the protein I was talking about, RHD3, now DER becomes different and is all trapped, it's ugly. And so what is the implication for plant growth? You can see this is the beautiful wild-type Arabidopsis. It's growing just fine, but the mutant has reduced growth. So when the endoplasmic reticulum does not function, does not look good, then we hamper growth. And this is major, because then we can understand how can we improve plant endoplasmic reticulum to make it more productive. And so that's something that my lab is contributing in understanding how can we force the endoplasmic reticulum to produce more and more proteins, for example. And so in the 80s and in the 90s, there was this blind approach of just pumping, literally genetically, the endoplasmic reticulum to produce more proteins. And this is the result. The endoplasmic reticulum will get fatter, as anything that does too much, and it gets flatter, and plants actually can die. So the productivity is reduced. So in my lab, we're trying to understand how can we go from a normal situation, for example, imagine the cars are proteins that have been asked, or that the endoplasmic reticulum is asked to produce, to a situation where we now have many more proteins, but the plants can cope just fine. So I'll get back to this in a second, but I just want to reiterate that plants can be forced to produce heterologous compounds besides trying to look after their own life and well-being. So I started the talk by saying that we have so many products that can come from plants. And I want to remind you all that the materials that we usually use besides the proteins from plants are stored or are made of the cell wall. So each cell, and this is a cell in the middle, is surrounded by this structure that is usually made primarily of sugars that is called cell wall. And why is this important? Of course, because we have materials for constructions, but also sustainable products like bed sheets, for example, or anything that you use normally and that you can also wear, like in cotton. So how can we understand how we can manipulate the secretory pathway to improve the plant cell wall and therefore plant productivity? So I just want to give you an example. Who likes jam? Who likes to eat jam? Yeah, me too, too much. Anyway, so jam, and if you like to do it too, you know that you had to add pectin, right? Why is that important? Because it makes it solidify. So pectin is a sugar and is produced by the Golgi apparatus, by some enzymes that are stored in the Golgi apparatus, and then is secreted to the cell wall. So in my lab, thanks to support from the Great Lakes Bioenergy Research Center, which aims to produce or to improve plants for the sustainable production of biofuels and bioproducts, we decided to try to understand how pectin is synthesized. And we came across a couple of proteins that I'm going to call CGR2 and CGR3, which have redundant functions, so they have very similar functions in producing pectin. But most importantly, they can modify pectin to make it more loose, okay? So now, by overexpressing, so overproducing these proteins, we can make a cell wall that is more loose because pectin is more loose. So what is the consequence of that? So let's take a look. Wild-type plants, this is Arabidopsis again, look normal, but when we produce high levels of CGR2 or CGR3, you can appreciate that these plants become bigger. And this is because we have loosened up the cell wall. So the cell wall is more plastic, and it can respond to an enlargement of the cell and allows the plants to expand. And we can take this trait from model plant species to trees, for example, like poplar. And we were able to demonstrate that poplars become bigger. So there is more biomass per unit of land. So we have made plants more productive. But not only that, we can actually make them more digestible. So we need less enzymes to digest the same amount of biomass of wild type. And why is this important? Because when we need to convert biomass to biofuels and bioproducts, you need a digestion of the biomass of the cell wall. So now we need to use 25% less of these enzymes needed to digest the biomass to have the same outcome. So this is a cost saving. But also, we can use these transgenic plants to feed them to animals because the biomass is more digestible, so animals can have a better outcome from their nutrition. And another example that I would like to make to you is mixed linkage glucan. So who had cereals for breakfast this morning? Most of you had jam, but only a few. Oh, this is not good. This is not good because if you eat cereals, you would eat MLG, or mixed linkage beta glucan, which is a mouthful on its own, but it's actually a very good sugar that is a fiber that allows improved digestion, but also reduces the risk of stroke. So I really encourage you to eat more cereals and drop that jam. So now, what's the problem? So what we're trying to do in my lab is to produce more of this MLG, because not only is good for humans, microorganisms can use it in fermentation to produce more biofuels and bioproducts. So more plants with more MLG, actually very good for the bio industry. So what we found out in my lab, but also in others, that when you try to produce MLG by overexpressing the enzyme that is responsible for its synthesis, F6, this is cellular synthase like F6, so I'm gonna just call it F6, and it's produced in the Golgi apparatus. You would see dots here showing where the enzyme is localized. When we overexpressed, unfortunately, the plants get sick. And this is due to the fact that we are producing an enzyme at very high levels that is first introduced in the endoplasmic reticulum and then goes to the Golgi. So we are intoxicating the endoplasmic reticulum. So in my lab, we developed a technology that is based on the overexpression of another enzyme that resolves the issues of too much protein being produced in the endoplasmic reticulum. And with this, we can now produce not only more MLG, we can also have plants that go back to the wild type state in terms of productivity. So we now transfer this trait into sorghum. So you can see that these are two transgenic plants. One is expressing one type of F6 from a model plant species that's called Brachypodium, and this is F6 from sorghum. And you can tell that these plants are identical to a control, but they produce high levels of MLG. And this is a finding that we got in the field, in field trials just here outside of MSU, where these plants are producing much more MLG. So I hope that these examples show you the power of working at the level at the subcellular level, how you can move findings from the lab into the field and produce plants that are bigger and definitely allow us to have a better outlook in the future. Now, our world population is growing. This is how I started. And I would like to say that in my lab, we're thinking bigger than our planet, and we want to explore new frontiers. We want to go to Mars eventually, if NASA allows us. So we've been very fortunate to work with astronauts on the International Space Station that have germinated our seeds. We send them wild-type seeds and seeds that have modified ability of producing proteins, and we were hoping to get genomic results on these plants to understand how can we improve plants to live in space. So our terrestrial seedlings, these are Arabidopsis again, this is how they look like. These are the seedlings that came back from space, and as you can see, you wouldn't even say that they're plants, but this is how, unfortunately, seedlings can grow on space. And obviously, there is so much that we are learning, and this is an example where we can push some of the frontiers. But last year, we were super fortunate to be given another opportunity from NASA and fly our seeds around the moon through the Artemis 1 mission, which is really setting the grounds to move science from the moon to Mars in the next, say, 50 years. And so the seedlings came back, seeds that we sent around the moon came back from space, and the seedlings are growing here at Michigan State, and we're checking how they responded to space flight. And I hope that if given a chance again to speak, I'll be able to give an update on those seeds. The project is really cool, and working with NASA is inspiring. So some take-home messages. I hope I convince you that plants are essential, and they are cool, and essentially that we can work with organelles to move from the tiny interior cell to the bigger scope, the bigger picture. And we need genetic engineering. And genetic engineering can allow us fast progress through modifying plants. And I'm really hopeful that plants can really allow us to explore new frontiers, new planets, and allow us to grow a sustainable life there. And all this would not be possible without the inspiring efforts of my team here at Michigan State and the funding that supports my research. So go green, and thank you so much. I put you all to sleep.
Thank you. Thank you, Dr. Brandizzi. There are a couple of questions that came in from online. And one is, tell us why organelles move in plant cells.
Oh, that's an excellent question. So the vacuole that I showed you on the movie occupies 90% of the cellular space. And so organelles have to move around this large vacuole to basically see the north, south, east, and west of the cell. And so they move around to make sure that they can communicate with the rest of the cell. As a matter of fact, if you reduce that movement, plants don't grow as well as wild type, because this movement is necessary for exchanging information and nutrients around the cell.
And the second one is, can we establish plant life on Mars?
I think so. We had to start from greenhouses first, but I think eventually we will be able in a very controlled environment. There is water there, so it is possible.
Thank you.
Thank you.
I think they've already grown potatoes on Mars, haven't they? Isn't it?
According to Hollywood, yeah.
So I thought the whole talk was really fascinating, Dr. Brandizzi, but I was particularly intrigued by the work that you showed very briefly about the antimicrobial peptides in plants. And so can you just say a little bit more about that? That's a really cool finding, I thought. So.
Thank you, Dr. Derrida. So essentially in plants, everybody thought until the work we did here that secretion occurs through the on motors, sorry, on the cytoskeleton made of actin. And instead in our lab, we discovered that the secretion antimicrobial compounds can happen through a machinery that depends on microtubules, and we identified the entire machinery that allows to do that. So there was a moment during the course of evolution where plants decided to go more towards the actin-driven organization in cells, and mammalians more towards the microtubule. And so I think the pathway that we have found really is the remnant of the last eukaryotic ancestor when the division of this labor was occurring throughout the evolution. Thank you so much.